Hello, hi students. I hope you all are doing well. So today we are going to start with chapter 2, that is structure of chromosomes, cell cycle and cell division. So in this chapter we are going to learn about the different parts of chromosomes, what does chromosomes consist of, the different phases in cell cycle, the different stages of cell cycle as well as the different phases involved in cell division. So let's get started. Now, any living organism is going to show this characteristic of division, that is cell division. So it is one of the most fundamental characteristic of life. And due to cell division, we are able to continue our life generation after generation. Okay, human beings and all the other animals are able to exist on the surface of Earth because of this process of cell division. So the cell division occurs in unicellular organism like amoeba, as well as very highly complex organisms like ourselves, humans, even the large organisms like elephants or the tall coconut tree. Each and every living organism undergoes cell division. Now, in cell division, first of all, the chromosomes get duplicated. जो भी हमारे बॉडी में जेनेटिक मटेरियल है वो पहले डुप्लीकेट होता है and it is getting evenly distributed into the daughter cells. Now what are daughter cells? जो नए cells form होते हैं उसे हम daughter cells कहते हैं. So this duplicated chromosomes are evenly distributed into the daughter cells and it is happening in a cell division called as mitosis. It is a type of a cell division. So mitosis में दो daughter cells produce होंगे. दोनों daughter cells को equal amount of chromosomes मिलेगा. So all the body cells, they are the result of a long chain of repeated mitotic division, and they are going to have same number of chromosome as any cells in a body. All the cells of the body are having the same number of chromosomes, and they are formed because of the repeated mitotic division. Remember, mitosis is a type of a cell division. So cell division is going to make sure that all the cells are functioning well. And if the cells are going to function well, of course, we are going to live a healthy life. Okay, it's going to lead to the normal functioning of an organism. Now, let us understand what are chromosomes. So, if we are going to take a normal living cell, which is not stained, it is unstained, and इस cell को अगर हम microscope के अंदर रखेंगे, under a light microscope, you will observe that its nucleus may not appear to contain any particular thing inside. Okay, अगर वो unstained है, and we are using light microscope, nucleus will barely show anything. But some cells, when they are dyed with suitable dyes, when they are stained with suitable dye, many structures that are present inside the nucleus are now noticeable. Can observe the different parts of nucleus now. So the nucleus is containing cells DNA. And this DNA is organized into discrete units called chromosomes. The DNA are arranged in a discrete units, which is called as chromosomes. Now, what do we understand by discrete unit? This is distinct units. So whatever DNA is present inside the cell, it is arranged, it is organized in a distinct unit, which is called as chromosomes. So what does the chromosomes consist of? It consists of one long DNA molecule, and this DNA molecule is associated with some proteins. Okay, we are going to learn about these proteins as well. So this complex of DNA and protein is called the chromatin. Okay, so DNA along with the proteins are called as chromatin. And when the cell is not dividing, you will observe the chromatin under the microscope, but जैसे ही cell divide होने लगते हैं, जो chromatin है वो condense हो जाते हैं, and it transforms into chromosomes. Okay, it becomes highly coiled structures and transformed into chromosomes, and this chromosomes can easily pick up the dye, and we can observe it under the microscope. When the cells prepare to divide, the chromatin fibers coil and they condense further, eventually becoming thick enough to be distinguished as separate chromosomes. And these chromosomes, they are going to pick up the dyes and they get colored. And that's why they are called as chromosomes, because chroma means colored. Okay, so these colored bodies are called as chromosomes, and they are highly coiled structures and condensed form of chromatin fibers. And remember, chromosomes are formed only when the cell is about to divide. It is preparing itself to divide. Now, let us understand who discovered the chromosomes for the first time. So, chromosomes in animals were first studied by a German scientist in 1882 by Walther Fleming. So, he noticed that there are rapidly dividing cells present in the larvae of salamander, and the microscope he used was quite an old type. And he saw some minute structures that appeared to be dividing lengthwise. So Fleming called their division as mitosis. Okay, so mitosis meaning thread. So he observed the thread-like structure that is actually the chromatin. And this chromatin, he observed, was dividing lengthwise. It is dividing lengthwise. And subsequently, with the help of more powerful microscope and some advanced technologies, different parts of the nucleus, chromosomes, are now being studied in great details. So we can observe in the diagram 2.1, this is a cell which is consisting of nucleus. Inside the nucleus, you all can observe there are chromatin fibers, right? It is present in the form of thread-like structure. Now, whenever the cell is about to divide, you can observe that now these chromatin fibers have begun to condense itself. And ultimately, it forms these chromosomes. So this is how the condensation of chromatin fibers occur, and chromatin fibers are converted from thread-like structures to chromosomes. Now let us see what is this chromatin made up of. So the chromatin material that constitute the fiber is made up of two substances. First is the DNA. Now DNA stands for deoxyribonucleic acid, which is present around 40%. It is forming 40% of chromatin, and remaining 16% is consisting of histones. It is a type of a protein. So chromatin fibers consist of DNA as well as it consists of protein, particularly called as histones. It's a particular type of protein and it forms around 60% of chromatin. Now, let's move on to the molecular structure of DNA. So the shape of DNA was first studied by Rosalind Franklin in 1953. But the structure, how does the DNA looks like, what we will be calling the structure as, was finally worked by Watson and Crick. They are the one who proposed the double helix structure of DNA. Okay, DNA structure को हम double helical structure कहते हैं, and this double helical structure was discovered by Watson and Crick, and for this they were awarded the Nobel Prize in 1962. Single DNA molecule is very large, and since ये बहुत ही large molecule है, that's why DNA molecule is called as a macromolecule. Macro means large. So the DNA is consisting of two complementary strands. Okay, now we will understand very soon why it is called as complementary. So ये दो complementary strands से बने होते हैं. There are two complementary strands and they'll run anti-parallel to each other. And they will be wound around each other in a double helix. So each single DNA is composed of repeating nucleotides. Now, what are the different parts of nucleotide? Nucleotide is composed of three components. Let's make a note of it. First one are the phosphates. Okay, DNA is made up of repeating units of nucleotides. बहुत सारे nucleotides मिलके DNA बनाते हैं. अब ये nucleotides किससे बने होते हैं? Phosphate, pentose sugar, which is arranged lengthwise, and nitrogenous base, which is attached to the sugar inwards. Okay, so what we have learned, DNA is made up of repeated units of nucleotide. बहुत सारे nucleotides arranged होते हैं back to back, and that forms the DNA. And what is nucleotide made up of? It is composed of phosphate, pentose sugar, which is arranged lengthwise, and nitrogenous base, which is attached to the sugar inwards. Now let us understand the structure of DNA with the help of this diagram. It is consisting of phosphate, sugar, and base. अब जो backbone होता है DNA का, it is made up of phosphate and sugar. Okay, they are arranged lengthwise. As you can see in this diagram also, phosphate, sugar, phosphate, sugar. It is forming the backbone. This is one strand. and this is the other strand. Okay, दोनों के backbone को sugar and phosphate बनाते हैं. And inside there is the presence of nitrogenous base. अब ये जो nitrogenous base है, दूसरे strand से, ये जो दूसरा strand है DNA का, उससे वो bind करते हैं with the help of hydrogen bond. Okay, so let us understand this nitrogenous base. So this is one nitrogenous base which binds with the other nitrogenous base of the other strand by the help of hydrogen bond. So let us understand the different types of nitrogenous bases. So there are 4 types of nitrogenous bases that are found in the DNA, which is adenine, guanine, cytosine and thymine. अब हमेशा adenine thymine से ही bind करता है, and they bind with 2 hydrogen bonds. Okay, as you can see over here, these 2 lines represent कि इन दोनों के बीच में 2 हाइड्रोजन बॉन्ड्स है. एडेनाइन और थर्माइन के बीच में 2 हाइड्रोजन बॉन्ड्स है. एंड ग्वेनाइन और साइटोसाइन एक दूसरे से बाइंड करते हैं with the help of 3 hydrogen bonds. So this 3 lines indicates that there are 3 hydrogen bonds between guanine and cytosine. Now remember, adenine cannot bind with guanine or cytosine. Okay, similarly guanine cannot bind with adenine or thymine. It's always adenine binds with thymine. Now, how you're going to remember this? Just remember at. Okay, adenine will always bind with thymine. Remaining, you're left with guanine and cytosine. They will bind together. So, as you can see in this diagram, this nitrogenous base is thymine. T is written, right? It is binding with adenine. And how many hydrogen bonds will be there between these two? So, there will be 2 hydrogen bonds. Similarly, we are having another nitrogenous base. Cytosine binds with guanine, and there will be 3 hydrogen bonds present between them. Again, adenine binds with thymine with 2 hydrogen bonds. So this is the basic structure of a DNA. And we can say that, we can observe in the diagram that DNA, there are composed of 2 complementary strands. Now, let's come back to the word complementary. It's complementary because adenine always binds with thymine, guanine always binds with cytosine. Okay, they are complementary to each other. And they run in antiparallel direction. They are made up of repeated units of nucleotides. See, this is 1 unit of nucleotide. Next, there is another unit of nucleotide. Next, there is another unit of nucleotide. So, बहुत सारे nucleotide, they are placed together and they form the DNA. So, they are formed of repeated unit of nucleotide. What does a nucleotide consist of? A phosphate, pentose sugar, as well as a nitrogenous base. Now, let's understand what are histone proteins. So, histone proteins are the proteins which is going to help the DNA in coiling and packaging into the structural units which is called as nucleosomes. Okay, so ये histone proteins help करते हैं DNA को in order to coil and pack. It will pack the DNA, and इस unit को हम कहते हैं nucleosome. So जो DNA strands है वो 8 histone proteins के around coil होते हैं. Okay, and this complex is called as nucleosome. That is, DNA along with 8 histone proteins forms the nucleosome. So an individual nucleosome can be imagined as a football around which long rope is wound around in 1 or 2 loops. And a human chromosome में there are millions of nucleosomes, and they are present back to back, and it is present in highly coiled form. Each human cell contains approximately 2 meters of DNA if it is stretched end to end. The human cells are having nucleus of around 6 micrometer in diameter. So how it is able to accommodate such a long DNA? So for this purpose, DNA is highly condensed in order to fit inside the nucleus. So this entire chromatin fibers is coiled and then it is supercoiled. And this supercoils, you can refer it to the coils of a telephone. And then it is able to accommodate inside the nucleus. So now let us look how the DNA are arranged around histone proteins and how it is coiled and supercoiled in order to form chromosomes. So we are having DNA over here. Okay. Then there are 8 histone proteins. As you all can observe, it is numbered also. There are 8 histone proteins around which the DNA is getting coiled. Okay. Once it is getting coiled, see, this single unit will be called as nucleosome. Similarly, there will be lots of nucleosomes that are being formed. Look over here. Okay. So these nucleosomes, they will start coiling and see how they are being supercoiled. Finally, it will form the chromatin fibers, and whenever the cell is about to divide, it will result in the formation of chromosomes, and it will be present inside the nucleus. Now, let us learn about the structure of chromosomes. जैसे ही cell divide होने वाले होते हैं, the cell is about to enter the cell division process. This chromatin is going to condense itself into chromosomes. Now, what does a chromosome consist of? It consists of sister chromatids. Okay, ये जो arms है, this forms, these are called as a sister chromatid. Okay, and a chromosome will consist of 2 sister chromatid. Okay, so this is the second sister chromatid, and both of them are joined together at a point of attachment, which is called as centromere. Centromere is a point of attachment. It is a small constricted area where the 2 sister chromatids are joined together. Now, it is not necessary that this centromere is exactly at the center of chromosomes. Now, there are different types of chromosomes based on the location of centromere that you will be learning in higher grades. Now, what is the function of the centromere? Now, this is the place where the spindle fiber will attach. It will attach the chromosomes. And as the cell division proceeds, ये spindle fiber condense होंगे and it will pull the sister chromatids towards opposite pole. Okay, it will break the sister chromatid and the sister chromatid will be pulled towards the opposite pole. And ultimately, दोनों daughter cells के पास एक sister chromatids पहुंचता है. After the completion of cell division, the chromatids, that is the chromosome, will now decondense. It will turn back into a thread-like structure, that is the chromatin fibers. Okay, so before the cell division, chromatin fibers present थे. जैसे ही cell divide करने वाला होता है, ये जो न्यूक्लियस के अंदर chromatin fibers है, ये condense होके chromosomes बनते हैं. Cell division में, mitosis particularly में, 2 डॉटर सेल्स फॉर्म होंगे. अब दोनों डॉटर सेल्स के पास एक सिस्टर क्रोमैटिड्स आएगा. Okay, and once the cell division is over, again this sister chromatids will decondense to form chromatin fiber. Okay, now remember, of course, the cell consists of various other organelles. Nucleus consists of nucleolus. So I'm not drawing all these things. I'm just giving you a rough idea how the chromatin fibers condense into chromosome and again how it decondense to chromatin fibers. Now, what are genes? So genes are the specific sequences of nucleotides. Remember nucleotide, the basic unit of DNA. So ये specific sequences है of a nucleotide present in the chromosome which will encode a particular protein. And जब ये protein express होंगे, it will result in a particular feature of a body. Okay, there is some particular protein that is responsible for your eye color. There is some particular protein that is responsible for the color of your hair, skin. Okay, so all these information is present in the genes. Okay, so genes are the specific sequences of nucleotide present in the chromosome that is going to encode for a particular protein. And when this protein is expressed, it will be resulting in a particular feature of your body. So इसलिए हम genes को कहते हैं units of heredity. And ये gene pass on होते हैं from the parent to the offspring, to the children. And it is responsible for some specific characteristic of our offspring. Okay, we often say that we have got this characteristic from a father or from a mother. It's actually the genes that are being carried forward from one generation to another. Let's talk about DNA fingerprinting. So there are lots of regions between the genes that are not at all functional. And this region make 99% of our total DNA. And because of these regions, there are tremendous variation from one person to another, and this helps in the identification of an individual, and that is called as DNA fingerprinting. So DNA fingerprinting, it is very useful in case of where the parent cannot be determined. So we can find out who is the father or a mother of a particular child using DNA fingerprinting method. Also, from the crime scene, when blood or semen stains are collected, hair or any item of clothing is collected, it can also provide a biological evidence by DNA fingerprinting. Now let's solve this progress check. Fill in the blanks. First one, chromatin fiber is made up of DNA and histone proteins. I hope you all remember DNA is making 40% of chromatin fibers and histone is occupying 60% of chromatin fibers. Next, the two sister chromatids of the chromosomes are attached to each other at centromere. Third one, the structure of DNA was first discovered by Watson and Crick. They were the one who gave the double helical structure of DNA. Next, the three components of the nucleotide are phosphate, then there is pentose sugar, which forms the backbone of the DNA, and the nitrogenous base, which is arranged inwards. Fifth one, let's move on. DNA strands wound around a histone octamer forms a complex called as nucleosomes. Okay, so nucleosome consists of DNA that is wound around histone octamer. And lastly, the question is, a specific sequence of nucleotides on a chromosome constitute a gene. Now let's move on and understand why there is a necessity for cell division. We have already learned in the beginning of this chapter that every living organism undergoes cell division. So what's the need for these new cells which are formed again and again? So new cells are produced for the growth of a body, to replace old and worn out cells, for the repair of old injured cells, and also for the process of reproduction. So let's look at each of these points one by one. Let's start with growth. So most of the organisms, plants, animals, human beings, we all begin our life from a single cell. That is a fertilized egg, basically a zygote. Okay, whenever a father germ cells, that is sperm, and the mother's germ cell, that is ovum, fuse together, it forms a single cell that is called as zygote. And we all start a life from zygote. Now, this cell keeps on dividing repeatedly. And ultimately, it forms a cluster of cells. You can see over here, there's a cluster of cell. Now, this cluster of cell will start taking shape. There will be formation of tissues and organs. And ultimately, there is a formation of an individual. So this cell division is very essential for growth. So see how we have started from a single cell and how the cell transforms itself to a human being. It becomes a fetus, right? So it is very important for the growth of an individual. So the next is replacement. So there is continuous wear and tear of our cells in a normal day-to-day activities. Like for example, 2 million red blood cells in a body are destroyed every second. And again, they are replaced by the new cells, which are formed by the division of the parent cells inside the bone marrow. In plants, there are old and dried leaves that falls off, and again, a new leaves grows out. So there is always a replacement of the old cells and it is replaced by the new cells. Next is for the repair. Now, apart from regular wear and tear in the tissues, there may be accidental injuries. Like one can get some cuts in the skin or there can be a fracture in the bone. So these cell division help in the repair of such injuries. So the cells will again divide, it will cover up the gap and it will even join the broken ends. So cell division is very important so that whatever injury our body is going through, it is being repaired. Now, in the cell division, usually for growth, replacement and repair, the number of chromosome, it will remain same. So the chromosome first will duplicate and it will equally distribute in the daughter cells. And this kind of division will occur in all the body cells, be it our cells of skin, liver, kidney, every body cells will undergo this type of cell division, having the same number of chromosomes, except for the cells that are involved in production of germ cells. Like, for example, oocytes in the ovaries and sperm producing cells, spermatocytes in testes. Okay, only these two places, the type of cell division is different. Okay, otherwise, in all the parts of the body, the cell will undergo mitosis. So, what is mitosis? It is a type of a cell division where the number of chromosomes remains same. So the parent cell is having 46 number of chromosomes. So daughter cells formed will also have 46 number of chromosomes. Such type of cell division where the number of chromosome is not changing is called as mitosis. And lastly, the cell division occurs for reproduction. So there is another type of cell division which is called as meiosis, and it leads to the production of sperms and eggs. Now, sperm and egg will only have half the number of chromosomes. So we will be learning about meiosis in details later in this chapter. But remember, there are some organisms like amoeba or bacteria, they can simply divide into two by the help of mitosis. Okay, but in higher organisms like humans and some higher plants, there is meiosis in order to reproduce. So what we have learned, all the body cells in our body is going to have equal number of chromosomes that is present in a parents. That is 46 number of chromosomes. Okay. And this 46 number of chromosomes occurs in pair. That is 23 pair. Half of 46 is 23. So 46 number of chromosomes occurs in 23 pairs. Now this state is called as diploid state. That is 2n. 2 into 23 will give us 46 number of chromosomes. Okay. So this state is called as diploid state. But if we talk about the sex cells, the sperms and the eggs, they are having only half the number of chromosome. Okay. So they are not having 23 pairs of chromosome. Instead, they have 23 number of chromosome. Only n number of chromosome. And such a state is called as haploid number of chromosome. Okay. Now, why is it so? Why the sperms and eggs are having only half the number of chromosomes? See, what happened is sperm contains half the number of chromosomes, that is 23. Eggs contain half the number of chromosomes, that is 23. Now, when they fuse together during the process of reproduction, it will result in the formation of zygote. Okay, so 23 chromosomes from the father fuses with 23 chromosomes from the mother. And ultimately, it will regain its original number. That is 46 number of chromosome or 23 pairs. That is, again, a diploid state. Right? All the human beings should have 46 number of chromosomes. So it is getting half from the father and half number of chromosomes from the mother. And ultimately, the normal number of chromosomes is maintained. Okay, now this number should not increase or decrease, otherwise it will result in genetic diseases. It should always be 46 number of chromosomes in our germ cells. So with this, we have completed what are chromosomes and what is it made up of. So in the next video, we will be learning about the different stages of the cell cycle. And if you all have any doubt, you can drop your questions in the comments. Thank you. Have a great day ahead.